Hi everyone, welcome to our new chapter, chapter 5. This one's going to be a fun one. It's all about DNA and chromosomes. Now, the content of the chapter is divided into three main sections. The first one is going to take a look at the overall structure of DNA. So this is when we're going to talk about the different nucleotide bases, the different types of bonds, the fact that it's a double helix. We're also going to talk a little bit, obviously, about its function. And then we're going to go ahead and move on to taking our DNA and how do we convert that into a chromosome, and emphasizing on a eukaryotic chromosome. What do they look like? How many do we have? And then last but not least, we're going to take a look at the regulation of the chromosome structure. And by then, we're going to take a look at things like histones and non-histone proteins and how they can be used to compact the DNA, and how certain sections of your DNA can be tightly coiled up, where others need to be loosely and easily available for things like gene transcription and translation. So lots of fun things to talk about. So without further ado, let's go ahead and let's take a look at some good old-fashioned DNA. So the first gift that your parents ever gave you was their DNA, right? So thanks mom, thanks dad. And it turns out that when we take a look at our gametes, the oocyte or the egg for the female and the sperm cell for the male, we see that each of them has the ability to carry their amount of DNA. And for humans, we're talking about 23 chromosomes. Now, mom gives you 23, dad gives you 23, and then at the end of the day, you end up with 46 chromosomes, or 23 pairs because they are diploid copies. And based on the DNA that you inherited, it will play an integral role in determining your overall phenotype. Your phenotype is anything from your physical appearance to some of your behavioral attributes, even some of the diseases that have a genetic link. They are all determined based on the genotype or the DNA that you initially inherit from your maternal and your paternal lineage. So it makes a lot of sense to know this molecule in detail, and obviously also know exactly how it can contribute to your body in many different ways. So let's go ahead and just do a quick little, I'm going to call it a review, because I know many of you have heard about the DNA molecule before, and I'm sure you already have a certain comfort level with it. So DNA stands for deoxyribonucleic acid, and in the name we can basically see that it's going to be composed of a backbone made out of a sugar and a phosphate, and then it will have a nucleic acid as its monomer, its building block, and that will be the combination of your different nitrogenous bases. So these are the letters that we think of when we think about DNA, right? We think about A, G, T, and C. Those are the adenine, the guanine, the thymine, and the cytosine. So those are going to be the individual building blocks of your DNA molecule. Now, if you go ahead and take a look at the illustration that is on the right-hand side, we can see that starting in A, we can see that the building block of the nucleic acid is going to be our nucleotide. And our nucleotide is basically a unit of the sugar phosphate backbone and one of the bases. So in this case, they added guanine. And if you combine those two, then now you have those nucleotides. And what we're going to do to create our DNA strand is we're basically just going to line up the individual bases, and the order or the sequence of letters will ultimately play a role in what genes we are able to express, and that of course will play a role in things like which type of RNA we're able to produce and what type of protein we're able to fabricate at the end of our transcription and translation detail. We also see that with the DNA strand that we can go ahead and describe it as a double helix. It is double-stranded. So for that, we can take a look at illustration C. And here we see that the sugar and the phosphate will form what we call the backbone. So you have one on either side. And then holding the backbones together or linking them together are going to be your bases. And as you can see from the pairing, we have this thing called complementary base pairing, which turns out that if we look at our four choices of nucleotides, adenine loves to pair with thymine, and cytosine loves to pair with guanine. They will use hydrogen bonds to hold on to their pairing, and if you look at your illustration, you notice that they have indicated for you that adenine and thymine will utilize a 2 hydrogen bond system, where cytosine and guanine will utilize 3 hydrogen bonds to hold the nucleotides together. Now, there is this thing called Chargaff's rule, and this was developed in the 50s based on when they started doing research on what the overall composition and structure of DNA was. And what Chargaff's rule says is that there's a balanced amount of nucleotides within each cell, so that if you know the value or the percentage of one nucleotide, you can easily figure it out for the rest. So what does that mean? So let's say, for instance, if I say to you that you're looking at a eukaryotic cell, and when we look at its nucleotides, 20% of them are going to have the adenine molecule attached to them. So then I'm going to say to you, well, what's the percentage that will have the cytosine attached to it? Well, that's an easy question, right? Because we know that if we want to go ahead and create the most stable form of DNA, we're going to have to follow our law of complementary base pairing. And that tells me that adenine loves to pair with thymine. So if I have 20% adenine, that will mean I have equal amounts of thymine. And that's what Chargaff rule is telling me. So if I have 20% adenine, then my T is also going to be 20%. So if you add those two together, you get 40%, which means that what's left over is 60%, right, of your sample. And that 60% is going to be equally divided between the cytosine and the, what's the fourth one? Guanine. You're correct. And because it's equally divided, that means that both of them are going to be 30%. There you go. Now, I know some of you will probably say, well, why didn't you just say if adenine is 20%, then the other pairing, cytosine and guanine, we can just do 50 minus 20. You can do that as well, whichever way works best for you. But either way, Chargaff's rule tells us that if you know the quantity or percentage of one, you can easily figure out for the rest because we're following our law of complementary base pairing. A goes with T and C goes with G. Is it possible for you to have a mismatch pair, A with G, C with T? Of course, but oftentimes if that happens, we see that the double-stranded DNA is very unstable and tends to fall apart and is at a high risk of passing on mutations. So our body will try to get rid of those by inducing maybe apoptosis within the individual cell with the mutation or the mistake. Now, another bond that we should mention when we take a look at our DNA molecule is going to be the bond that actually keeps the sugars linked to each other within the backbone. And that is going to be a phosphodiester bond. This is an example of a covalent bond, and it's going to involve the 3' sugar bonding to a 5' it involves the binding of the 3' carbon on one sugar to the 5' carbon on a second sugar. My apologies for that. And within the 3' we see that there tends to be a hydroxyl group, so an OH group. Whereas on the 5' end, we tend to see more of a phosphate group, PO3. And the interaction between those two is in essence what's going to allow us not only to hold the backbone together, so this is what's linking together the phosphate and the sugar, but it's also going to be responsible for producing the polarity of the overall DNA strand. DNA has an overall negative charge, and as you can see right here, it says to you, depending on the chemical differences that's created by the ester linkage between the 5' carbon and the 3' carbon of another sugar, it's going to give rise to the polarity of the resulting DNA strand. So the interaction between the 3' and the 5' hydroxyl and the phosphate group, it's what's responsible for producing the negative charge of the DNA molecule. Now, it turns out also the reason why the 3' and the 5' are often selected has to do with its overall orientation for stabilizing the basis so that A can bind with T and G can bind with C. But also when we take a look at their overall structural view, the 3' sugar is often described as looking like it has a little hole, where the 5' is often described as having a knob. So the knob end of the 5' fits perfectly into the whole end of the 3'. So it all really has to do with structure and orientation and allowing it to fit most comfortably. But at the end of the day, we're going to walk away with a phosphodiester bond, which is going to stabilize the backbone of the DNA molecule. And then the rungs, the inner bases, the A, the T, the G, and the C, those are going to be utilizing their hydrogen bonds to link together the double helix. When you look at the overall structure of your bases, the adenine, the guanine, the thymine, and the cytosine, you're also going to notice that they're often classified as either a purine or a pyridine. So a purine or a pyridine. And this is based on their chemical structure. A purine means that when I go and I draw the structure, I'm going to notice that it has a double ring structure. So go ahead and take a look at your picture. Here's my guanine. And here's my adenine, and both of those are classified as purines because they have two ring structures. So here's one, here's two, here's one, here's two. So purine means I have two rings. The pyridine only has one ring, so that's going to be my cytosine, here's my one, and my thymine. In case you were wondering about uracil, uracil would also be considered a pyridine because it only has one ring structure. So here you can see that if we're going to do our complementary base pairing and utilize our different hydrogen bonds to keep our nucleotide bases together, you're going to notice that the best thing to do is, in addition to pairing up A with T and G with C, is that you're pairing up a purine with a pyridine. A purine with a pyridine. And that turns out to give a nice stable formation between the internal rungs of the DNA molecule. Now take a look at your right side illustration. So this is B. And then notice the interaction between the phosphodiester bond forming the backbone in yellow. So that's the interaction between the 3' sugar and the 5' sugar. And then in the middle part, you can see your bases. And then notice with the red dashes, those are going to be my hydrogen bonds that are keeping together what they like to call the rungs or the internal workings of your DNA molecule. Another thing that I want to point out is that it turns out that if we really want to stabilize those two hydrogen bonds between the adenine and thymine and those three hydrogen bonds between the guanine and the cytosine, the best thing to do is to take our phosphate sugar backbone and allow it to run in what we call an antiparallel format. And antiparallel basically means that on one side of our double-stranded DNA molecule, our backbone will run so that, for instance, the 5' carbon is facing upwards and the 3' carbon is facing downwards. Okay, so that will be the orientation of the first phosphodeoxyribose backbone. The other side of the double-stranded DNA will run the opposite way. It will run antiparallel, meaning that the 3' sugar will face upwards and the 5' sugar will face downwards on this end. So as you can see, they're antiparallel because it's 5 on one side, 3 on the other, 3 on one end, 5 on the other. And the fact that we know these little characteristics about the DNA molecule then allows us to kind of predict what a daughter strand of DNA will look like if we know what the initial piece looks like. So for instance, if I say to you, I'm looking at one piece of DNA and it starts 3', and then I see that the nucleotide bases that are being held together in the middle are in order adenine, guanine, cytosine, thymine, and then it ends, of course, with the 5' carbon pointing downwards because we know 3 on top, 5 at the bottom. So if I say to you, I know one piece of the DNA, what does the second piece of DNA look like, right? Because it's a double strand. What does the second piece look like? How would you determine that? Well, the first thing you have to remind yourself is that the backbone is going to run antiparallel. So if on the one that I'm giving you, the backbone starts with a 3 and ends with a 5, that means that the strand you're looking for is going to start the opposite. So start with a 5 and it's going to end with a 3. So that'll give you your antiparallel format. Now for the letters, you're going to remember that they're being held together by hydrogen bonds and they're going to do their complementary base pairing. And that basically tells you that if I have adenine on one side, I'm going to have thymine on the other side. That's linking it together. If I have guanine, it's going to be followed by cytosine on the other hand. And if I have cytosine, it's going to be followed by guanine. Here's another cytosine, so there comes another guanine. And last but not least, if there's thymine on one end, it's going to be followed by adenine on the other end. So if you know what one side of DNA looks like, and you keep in mind that the sugar backbone runs antiparallel, and you keep in mind that the letters all have their complementary base pairing because they're following that rule, and we're relying on Chargaff's rule as well that tells you that everything's balanced, you can easily then dictate or predict what the other strand of DNA is going to look like. Also, what we see happening is that because of the antiparallel formation and because of the overall interaction of your bases, we see that when DNA coils up, it tends to coil up more in what we like to call a right-handed turn. And as it's forming its right-handed turn, it starts to create the space-filling model that you see right over here on the right-hand side. And you see it starts to form a double spiral, kind of looks like a little spiral staircase. Someone once told me that it kind of looks like pearls coiled up together under the electron microscope. But either way, what we see happening is that as it's coiling up in the right-hand turn, this is its most energy favorable position. There will be about 10 base pairs per turn. Each turn will also have two grooves. It will have a major groove and a minor groove. And the reason I like to point out these grooves is because oftentimes when we take a look at how water molecules are going to be interacting with our DNA strand, they will do that interaction either through the major groove or the minor groove of the molecules. This is a right-handed turn. I do want to say that there's also a left-handed turn. That one is not as energy favorable, and that one creates more of like a zigzag pattern when you line up the double-sidedness of the DNA molecules. So both right-handed and left-handed turns are seen in the body, but the right-handed turn is by far the most common version of stored or accessible DNA. So now that we've refreshed our minds about DNA, deoxyribonucleic acid, and A goes with T, G goes with C, using hydrogen bonds, and you have your sugar phosphate backbone with the ester bond, let's go ahead and see how DNA correlates with the term genes. So genes, by definition, are simply a segment or a certain sequence of your DNA. And the reason that sequence is so important is because they will basically have the ability to do things like transcription and translation. So on your PowerPoint, I wrote down for you that genes will mostly code for proteins, and that will allow us to have what we call gene expression, which is that you do the whole process of transcription when you go from DNA and convert it to RNA. And translation, which is now when you take your RNA, like messenger RNA, and you utilize the help of ribosomal RNA and tRNA, transfer RNA, to ultimately produce a protein in the body. And as we've previously discussed in chapter 4, the proteins are really the workers in the cell, so our body produces hundreds of thousands of them to maintain our homeostasis, produce our various secretions. Anything you can think of it, usually it involves a protein. It's important to point out that not your entire DNA strand has genes. So not your entire DNA strand from beginning to end is able to do transcription and translation. In fact, within our DNA, we have what we like to call genomic sections and non-genomic sections. Genomic sections are the ones that house the genes. So those particular formulations of A, T, G, and C will determine which RNA and which proteins are produced. The non-genomic sections are often utilized for stabilizing the genomic section. They can often also be used as evidence of evolutionary transfer from one generation to the next. We even see that within certain closely related species that they'll have highly conserved non-genomic sections embedded within their DNA. So it seems to play a role in the overall stability as well as assistance of the replication of the DNA strand. Non-genomic DNA often used to be called junk DNA, because for a long time we believed that it just kind of sat there, it didn't really do anything, it didn't contribute to the system. But it turns out that the more we learn about the DNA, the more important this junk DNA becomes. So therefore, we now like to refer to it as more of a non-genomic DNA. It still plays an essential role within our overall stability of our DNA molecule. In case you're wondering, the latest article basically determined that if we look at our entire DNA strand, all of our nucleotides, we have about 98% of our DNA that is classified as junk / non-genomic DNA, and there's only 2% of our DNA where we actually house the genes that produce all the proteins that we rely on for our genomic expression. In fact, humans have about 2,400 genes, and those 2,400 genes are able to produce hundreds of thousands of different proteins. Before the Human Genome Project, scientists widely believed that each gene was only able to secrete one protein, but now we know that the genes are able to do things like alternative splicing, which means that when they secrete, when they do their RNA molecule, there are certain sections called exons that they're able to splice out and rearrange, and depending on how the exons are arranged, that will ultimately determine the codons or the sequence of the messenger RNA, which will allow for different proteins to be expressed. So it's kind of like taking little blocks and rearranging them, and as you rearrange them, your sequence of nucleotides changes, which means your directions change, and thereby your end product will change. So one gene has the ability to produce several proteins. We will talk more about transcription and translation when we go into chapter 7, so there's a lot of fun to come, and you'll even have an opportunity to take a DNA strand and convert it into a protein or a polypeptide sequence utilizing the different steps of transcription and translation. But for now, I just wanted to remind you that a gene is a section of your DNA that's able to be expressed in the form of proteins, and that transcription is the first step. That's when you take your DNA and convert it to RNA. And the reason this step is so important, especially for eukaryotic organisms, is because the DNA that's being housed in the nucleus is not able to exit the nucleus. So therefore, it has to make a copy of its specific genomic section, and that will then become the RNA, which is able to go in and out of the nucleus as it pleases. And it's able to go into the cytoplasmic region where it's able to do translation, meaning it converts itself or it messages itself and produces proteins depending on the amino acid sequence that it is coding for. In case you're wondering about the length of your DNA, we have 46 chromosomes altogether, and that basically contributes to about, or accounts for about 48 million nucleotides. So 48 million adenine, thymine, guanine, and cytosine collective. That's a lot of nucleotides to be housing in those tiny little cells. So based on the overall composition with the nucleotides, because it's such a massive amount, what we see happening is that it's very important for our cells to be able to condense or compact DNA down as much as possible. So something we have to talk about is how do you go from having a piece of naked DNA, so a piece of just double helix, double-stranded DNA, how do you get that piece of DNA to coil up so tightly that it gives us these beautiful tiny little chromosomes? And in eukaryotic cells, the chromosomes will often have a linear format. In bacteria cell, in the prokaryotic cells, bacteria tend to have one chromosome. That chromosome tends to have a circular format. And don't let the one chromosome fool you, because it can still do plenty of functions. Bacteria cells have been around a lot longer than we have, and they are thriving. It is also very common for the bacteria to have multiple copies of that one chromosome floating around in their cytoplasmic region. But one of the big differences is that it has a circular chromosome, whereas we have more of a linear chromosome. Now the chromosomes for humans, as I said to you before, we have 46 altogether. or you could say we have 23 pairs, and out of those 23 pairs, 22 of those are what we like to call autosomes. Auto means it's the same in both male and females. And then our second pair, our 23rd pair is going to be our sex chromosomes. And that, of course, from a genetic standpoint, is going to determine if you are female, which means you inherit both X's, one from mom, one from dad, or if you are genetically a male, which means that mom gave you an X and dad gave you a Y. And I could say that because the male species is the only one that carries the Y chromosome. So if you think about it, the parent that determines the gender of the children will always be the father, because mom has 2 X's, so that's all she can contribute, where dad has either contribute an X or a Y. This, of course, is a reminder that humans are what we like to call diploid, 2N. We get 2 copies of everything, one from mom and one from dad. When it's time for us to produce our gametes, our sex cells, so the sperm and the oocyte, we take that diploid number and we half it, and those cells become haploid. That's supposed to be an N, meaning that instead of carrying 46 chromosomes, it only carries 23 chromosomes. And that is then the initial gift that your parents gave you, which is how we started off the lecture. The fact that dad's sperm gave you 23 and mom's egg gave you 23 to collectively create a fertilized egg, aka a zygote, that has 46 chromosomes. Because for us, the magic number is always going to be 46. That is where we would like to come to. That is when our cells function the best. Now, you can easily use the term genome to describe all of our DNA collectively as a whole. And within our genome, if we want to visualize it, we can go ahead and we can create a karyotype. And a karyotype basically means that we take our cell, we lyse the nucleus, and we fluorescently stain, or through the addition of color coding after the picture's taken, we're able to fluorescently stain or indicate all the different chromosomes that are lining up. Now, a karyotype will also often take it further by pairing up the chromosomes. So it will find the maternal pair and the paternal pair. It will pair them up and it will organize them from tallest to shortest chromosome. So go ahead and take a look at figure B, and you can see a beautiful karyotype for you right over here. You're going to notice that all the chromosomes are paired. So thanks, mom. Thanks, dad. Thanks, mom. Thanks, dad. Thanks, mom. Thanks, dad. Because each of them gave you one version of that particular chromosomes. They are lined from tallest to shortest. And you're also going to notice that they're numbered 1 through 22. Those are going to represent our autosomes. And then the 23rd pair does not get a number. Instead, those, because they are our sex chromosomes, are either going to be indicated as having 2 X's or 1 X and a Y. So this right here is a karyotype of a female, a genetic female, due to the fact that she has 2 X chromosomes. The karyotype is a picture taken during metaphase, when the chromosomes are aligned down the center of the cell, making it easier for them to be seen in their paired format and in such a linear formation. Oh, another thing I wanted to mention, my apologies, homologous chromosomes. So homologous chromosomes refer to the fact that you have a contribution from both your maternal and your paternal gametes, and because you have that, we see that you have 2 of each chromosomes, making them homologous, but it does not make them identical. So let's talk about that a little bit more, and let me go ahead and show you a picture to kind of showcase what I mean when I say homologous chromosomes. All right, my apologies. We're going to hold on for the homologous for just one more second. I quickly wanted to mention, and we're going to talk more about this in the second part of our lecture, how you're able to take your naked DNA. So if you start on top of this picture right here, this picture comes from a McGraw-Hill book, but it basically shows you a nice way of how you can go from least compact to most compact. So on top is my naked DNA strand. You can see my sugar phosphate backbone. You can see my hydrogen bonds. You can see my nucleotides. interacting with each other. And what we're going to talk about is how do you go from this to this? And that basically means how do I make a chromosome? Because the chromosome is going to be my compacted version, my most compacted version of my DNA strand. It's essential that this process gets done because there's over 6.5 foot of DNA in every single one of our cells, which means there's a massive amount of DNA that we have to coil up very tightly. So part of what we see happening is that the DNA molecule will start interacting with these proteins called histones, and it's going to allow it to wrap the DNA tightly, creating this process called nucleosomes. And we're going to discuss this in a lot more detail in the second part of the lecture. But I want to show you how the nucleosomes will stack together and they're going to create chromatin. So chromatin is coiled up nucleosomes. And nucleosomes, it's basically the addition of DNA, naked DNA, wrapped around proteins. And I wanted to introduce the term chromatin to you because you're going to see that all over the place. So whenever you see chromatin, I just want you to think coiled up DNA, coiled up in the form of nucleosomes because it wrapped itself around histones. And the chromatin is going to stack together and condense down further and further and further until it gives you the beginnings of your chromosome structure. Now on this illustration at the bottom, notice how it says a replicated chromosome. And that's because we're going to talk about the fact that during the cell cycle, there will come a time where you have to replicate the DNA that you have, where you have to go from 46 to 92 pairs. So when you replicate the chromosomes will hold on to their copied version, and that copy version is what we're going to call a sister chromatid. So see, TID. So chromatids are replicated sections, and chromatin, TIN, is coiled up DNA. And the sister chromatids, the duplicated sections, are going to be linked together in the little centromere, the middle section of the chromosome. And it's going to hold on to the sisters until it's time to divide, and each of them will go their own way. I'll give you a quick little overview of the cell cycle in our next discussion, but you'll be able to see how the chromosomes will pair up and then they'll detach from each other so that we can create new daughter cells, a next generation of cells. So once again, if you see the term chromatin, TIN, that simply means that it's condensed or coiled up DNA that's been surrounded or wrapped around histones, making nucleosomes. And as the chromatin replicates itself, we then call the replicated sections chromatids, TID, sister chromatids, and that will then be held together by a little centromere. And in fact, the replicated chromosomes in metaphase is what you come across when you take a look at your karyotype map. Here's the picture that I wanted to show you regarding the homologous pairs. So in this picture, which I also stole from a McGraw-Hill book, we see a representation of your two sources of DNA. Using typical colors for genders, which is a whole other discussion. Either way, it shows you right here. In pink is meant to represent the chromosome that you inherit from your maternal lineage. So thanks, Mom. And then in blue is going to be the chromosome that you inherit from your father, the paternal lineage. Now, it turns out that each of them is going to give you 23 chromosomes, right? So you're going to end up with a diploid number, 2 of each. So let's assume that this is chromosome number 5. Okay, so Mom will give you 1 and Dad will give you 1. Because you get 1 from 2 alternate sources, we're going to call it a homologous pair. Both of them are chromosome 5, but not all of them will be identical. Not everything in that chromosome will be identical between the paternal and the maternal match. What do I mean with that? Well, go ahead and look on the inside of your chromosomes and you're going to see the letters A, B, and D. So A, B, and D. Those are meant to represent my genes. And it turns out that genes come in different variations, aka alleles. The version that you inherit from your mother isn't necessarily the same as the one you're going to inherit from your father. So this is when we start thinking about things like some genes are dominant, meaning that they're always expressed. Others are recessive, which means that they can be masked or hidden. So you can have different alleles or different variations of the same genes, and that is then why when you come across the term homologous chromosomes, I want you to think about the fact that you have two separate origins. They come from two different sources. They will not have the same version of all of the alleles. So as you can see in my example, for instance, mom gives you the dominant version of gene B, indicated by a capital letter, where dad is giving you the recessive version of that gene. So then we would have to go ahead and take a look at things like our phenotype to figure out if it's going to follow a traditional rule of dominant being expressed and recessive being masked, or if there's another rule in the genotypical expressions. Genes don't always follow the dominant and recessive rule. But because they come with two different alleles, two different versions, they are not 100% identical. So please keep that in mind. That is also one of the reasons why offspring will have genetic variation based on their parental strand, because obviously each parent is only able to contribute half of their genome. And in addition, what we see happening is that within the half that they donate, there are lots of variations of the genes that get carried over from either the maternal or the paternal lineage. In this illustration, they also indicated for you the fact that you have your sister chromatids. Remember, sister chromatids just means that one side is replicated and being housed together by the little centromeres. Okay, so homologous pairs of chromosomes means that you have the same chromosome from two different sources. In case of us, because we're diploid, we get it from two different sources. But the genes that are being expressed on the chromosomes do not have to be 100% identical, because it will all depend on which version of the gene or which allele you inherit from your parental source. I also wanted to show you some examples of how we can use a karyotype to detect any type of abnormalities within our patient or within a source. So these are all examples of human patients that came in that had some abnormalities within their genome. So the one that's on the left, if you can take a look, you can see that you have the chromosomes nicely paired up. They're numbered 1 through 22, organized from tallest to shortest chromosome. And then right here in the corner, you can see that you have 2 X's, no Y's, so that means that once again, we're looking at a genetic female. Now, the abnormality in this karyotype, can you see it? It is right over here. If you take a look at chromosome 21, you're going to notice that instead of it being a diploid, it's actually a triploid. This is called trisomy 21, and this is what we see in a lot of patients that have Down syndrome. Down syndrome is a genetic disorder where the patient is born with 47 chromosomes instead of 46, and in a lot of times this will happen with chromosome 21, where it will be able to house a third copy of itself. I do want to note that a lot of times from an embryonic development, if there's variations within the chromosome number, so if you would have like 47 or 45, it will cause what we like to refer to as spontaneous abortion. The oocyte, or I'm so sorry, the zygote, the fertilized egg, will stop developing. It will induce partial apoptosis, and the egg will not attach itself or loosen up, and it will basically be ejected from the female's body, just like she would have a normal menstrual cycle. The spontaneous abortion usually occurs very early on, within the first few days of development, and part of that has to do with the fact that our body is basically innately set up to reject any number besides 46 chromosomes. As you can see, there are some that are able to defy the odds, and the cases of Down syndromes are very interesting because we see that chromosome 21 is able to allow for a third copy of extra DNA. So that's why that zygote is able to continue its development. There are also some examples where you can have abnormality, or what they like to call non-disjunctions, meaning uneven splitting of the sex chromosomes. And for one example of that, you can go ahead and take a look at the karyotype that's on the right. And once again, you can see that all the chromosomes are paired up and they're organized 1 through 22. There's nothing wrong over there with the autosomes. But come over to the sex chromosomes, and what you're going to notice is that there's only one X, and there's absolutely no Y. So remember, if this was a female, you would have 2 X's, and if it was a male, you would have 1 X and you would have 1 Y. So the karyotype that we're looking at over here has 45 chromosomes instead of 46. And in this case, what we see happening is that it's missing a sex chromosome. Now, this is what we like to call Turner syndrome. And in Turner syndrome, you're looking at an individual who's genetically a female, because as soon as they have the X chromosome, they are designated female. Female is the default sex of humans. And the reason that is because your X chromosome basically has all the genetic information needed to develop from zygote all the way to baby or fetal stage. The Y chromosome basically is used for what they like to call the sex determining region, which basically says, hold up, we need to have more testosterone, we need to do the male reproductive system, we're having a boy. And that's one of the reasons why the Y chromosome is very small compared to the X chromosome. It's just basically there to say, nope, not going to be a female this time, it's going to be a boy. Whereas the X has all the developmental genes needed. So what we see happening here is that with Turner syndrome, because the oocyte still has access to one X chromosome, it is able to develop. Children with Turner syndrome, they basically will see later on when it's time for puberty that they will not develop their menstruation cycle. They'll have a shortened height. From a mental aspect, usually there are no issues. That means that the child can developmentally be perfectly normal. There are some phenotypical abnormalities besides the short stature. They will also not fully develop their secondary sex characteristics, so the development of the breast is delayed. They also tend to have a thicker neck than normal. And oftentimes, because they have a delay in their reproductive system, they're also very infertile. And part of that is because they have an odd number of chromosomes that cannot be evenly divided successfully in their gametes. So this is an example of Turner syndrome. And in case you're wondering, is it possible to only be born with a Y chromosome, meaning you have 45 chromosomes, meaning 22 pairs are autosomes, and for your sex chromosomes, you just have the Y, and it turns out that has never been recorded, and part of that is because you would simply be missing too much genetic information, because remember, the Y at the end of the day just says it's going to be genetically a boy, whereas the X says you're going to be genetically a female, but it also gives you instructions on the development pattern. Now, you don't need to necessarily have an abnormality in, abnormalities are not limited to the total number of chromosomes. Sometimes you can just have an abnormality within one chromosome itself. So that's why I also wanted to include these two pictures. The picture on the left, as you can see with the boxed in by the X, is showing what we like to call Fragile X syndrome. Fragile X syndrome is a genetic disease where we see that the X chromosome gets added on nucleotides, becoming much longer than normal. And this Fragile X syndrome, as it's passed on from one generation to the next, the appearance of the first symptoms will actually occur earlier and earlier as it gets passed on longer from generation to generation. Fragile X syndrome usually is correlated with a lot of different learning and mental disabilities, and it's all due to the fact that you have that extra strand of DNA that's by the X chromosome. The longer the access strand is, the earlier the symptoms seem to be setting on. The picture on the right is that of what we like to call a translocation. Some of you might call it a jumping gene, but as you can see in this particular picture, if you go over to your chromosome 15, you're going to notice that when you look at the pairs, one is longer than the other. And if you go over to chromosome 22, you're going to notice that there's only one set of it. What happened to the second piece of chromosome 22? Well, it turns out that it translocated over to chromosome 15 and it attached itself right over here. Go ahead and take a look at the insert picture. You can see right here the length of the missing chromosome fits in perfectly with the axis of the additional length in chromosome 15. So we see that this happens. We're not really sure why it does. They like to call it jumping genes, meaning that the genes basically jump from one side to the next. However, the more scientific term for it is called translocation. This is an example of what we call unbalanced translocation because one side gets longer and the other one gets shorter. There are also examples of where you can have balanced translocation, and that usually means that one side will swap out with another non-homologous chromosome, but they'll equally restore the original size. So for instance, I can take a little piece of chromosome 1 right here and swap it out with the non-homologous chromosome number 2. And if I swap it out in a way that the end result is that I still have the original length of chromosome 1 and chromosome 2, I would then make it a balanced translocation. Whereas in this example, I have made one longer and the other one shorter, so therefore I call it an unbalanced translocation. Another thing that I definitely want to mention, and this kind of comes into play if you start taking a look primarily at closely related species, is that oftentimes you'll see a wide variety in the amount of DNA that they're able to house. You will also see some differences in their overall chromosome number and chromosome formation. So right here you have two closely related deer species, the Chinese deer on the left and the Indian Munchak. I know I'm mispronouncing that, but it's a deer species, so excuse me. Either way, what we see happening is that the Chinese species has an older lineage. The Indian one is a little bit younger. So if we compare the DNA, what we see happening is that they will go ahead and notice that a lot of the chromosomes that were originally found in the older species, the Chinese one, we can also locate them in the Indian one, but their shape has completely changed due to the fact that a lot of the individual pieces from the Chinese lineage have actually fused together to create larger chromosomes. But when we take a closer look, and you can't really see this from the picture, if we take a closer look at the number of genes and their overall expression of their phenotype, we really don't see that there's a major change. So it's just all about how the DNA is compacted. The genes are still accessible between both species, allowing for very similar phenotypical expressions to occur. One last thing that I want to add to part 1 of our lecture right here is that just because you have a large genome doesn't necessarily mean that you have a lot of genes to express or a lot of proteins to produce. So the more DNA doesn't necessarily mean a higher order of development. It will all have to do with how much of the genomic sections you have that are being able to express. So there's lots of examples with organisms that will have more DNA than humans. Excuse me, but because of the simple fact that within that massive amount of DNA, they don't have as many genes as we, they're not able to produce such a wide variety of proteins that we are able to do. So the size doesn't always matter. It's all about how many genes you have and how you're able to express them. Alrighty, so let's stop over here, and then when we meet up again for part 2, we'll take a look at duplication of DNA and then how we can modify those chromosome structures.